and just officially bid you very welcome, Georg. I'm trying to pronounce this German name correctly.
I
And
was going to ask, is that the right way to say it?
It is perfect. Anything
Gee,
that starts with a G is fine.
I'm going to stick to George.
That's perfectly all right.
Good old-fashioned British way.
yeah, I think a Danish enculturation on my side helps a little bit with the German pronunciation. But
You're doing it properly.
and uh, just as a brief reminder, we usually do a presentation, then we have some time for discussion and feedback. Usually, Carl summarizes in the end. We're not so many people today, so if Carl or Thomas wants to change things up, they will let me know. But otherwise, we have that default that we usually follow.
Yeah, just to say, this is an unusual ad hoc TMB meeting, and my thanks to George for participating. There's an interesting, just to say, in the past 2 weeks, I've had to review two mammoth reviews of predictive coding, and none of us were on the co-authorship. There was one massive authorship. I think there were about 20 co-authors in some open science initiative. And they'd submitted, I think it's 125 pages reviewing predictive coding. Then I got another one from one of our erstwhile colleagues in Germany, reviewing things from their perspective. So this is clearly quite a hot issue, and you know, it did strike me that there's a lot of interest in, presumably, George, the community, these authors were largely electrophysiologists. The second one was had more sort of systems neuroscience, brain imaging people involved, but certainly the perspective they were taking was deep into the weeds of the sort of the microcircuits and the electrophysiology. So I'm looking
Yeah, I
forward to
think
this. So hearing it from your side of the fence as opposed to the maths.
perfect. Yes, that was precisely my idea, that it would give you a sort of an overview of at least where my thinking comes from and what the key questions were that we were trying to address over the last 2 years. Should I get started then?
I think so.
Let me share. And given that we're so a small crowd, please interrupt at any moment. Sorry, I'm now being incompetent. There we go. Does that work?
Yes, we see a
Very
screen.
good. Very good. I just realized the title's wrong anyway. Right. So just to give you the way I approached the question in general was through imitation learning. I mean, people understood for a while that songbirds can learn to imitate, so can humans, obviously, sounds. And the question is, how does that work? And at the time we discovered, people were thinking in very different terms about this, more in terms of reinforcement learning. But then we discovered serendipitously that there's these error cells in the auditory part of the bird brain. Conceptually, we thought about it in this way. If you imagine that there's brains that are composed of sensory regions and some effectors, the task that you want to learn is a mapping from sensory space onto motor space. This in the terms of oh, well, sorry. Are you still there? I'm sorry, I just locked my computer instead of getting the laser pointer.
We can still see and hear you, but the screen is black.
Very
Yeah,
sorry.
it works.
I'm back. Yes, the old Windows L versus Control L error. I'm so sorry. Right. So if we had the inverse model is difficult to learn. This is an old internal model idea that came out of the work from Jordan and Rommelhart and Daniel Wolpert and Kawato and colleagues. Anyway, and they noted that it's much easier to learn this forward model, which is the inverse map, and then you can train the whole thing as an identity map, which makes it easy to compute or to train the inverse model. Anyway, based on this, the reason this is easier is because you can cheat. You have the world, of course. I mean, you're all well aware of this. But this was our sort of thinking, right? And the old terminology, this is how I usually introduce it, this corollary discharge, reference copy, learning signal in predictive processing just translates to sensory prediction and prediction error. The key is that the key prediction of this idea at the time, and we didn't realize how close this was to predictive coding or predictive processing when we started, is that there's prediction error signals in visual areas or sensory areas of the brain. Right? And the experimental strategy for everything we've always done is to break the coupling with the world. Right? Simply because movement, right? This relies on the assumption that movement is likely the best predictor of any sensory input an animal has, simply because of like the evolutionary history. Nevertheless, most physiology labs tend to study this using oddball stimuli or other learned associations. Right? We have always relied on this coupling between movement and visual feedback. Now, right. So what we did then is just write down a hypothetical minimal circuit. I mean, I'm a circuit neuroscientist, so I like to think in how is the computation actually implemented. Right. The simple one is how do you compute the prediction error, and very basically this is just a subtraction of two signals, some sensory signal and a prediction of that sensory signal, and you can write this down, right? Either mathematically, so the way Round Ballard did, or in circuit neuroscience via only one way. There's many ways to do this, but one would be just a subtraction. Circles are inhibitory neurons, triangles are excitatory neurons. And because these signals, the excitatory neurons cannot fire below zero, right? This is a subtraction S minus P, but if P is larger than S, this will give you a negative number. So a simple trick is, we argued at the time, a simple trick is to just split this across two neurons to compute this, basically a rectified version of this subtraction. And then just to make this work, obviously, you're probably familiar with this, you need then these internal representation neurons that integrate over these prediction error responses. And this isn't obviously our idea. This is just a circuit version of the Round Ballard idea. There's one detailed difference that I'll speak about briefly later. But that in essence is the idea. Can we put now can we identify which neurons these are, right? What neuron, where in the cortex does this positive prediction error sit? Which type of inhibitory neuron is this that receives a top-down prediction and sends it on to the positive prediction error neuron, the same for the negative prediction error? Are there genetic labels? Can we identify them in cortex? Obviously, I mean, that's what we spent the last 10 years doing. And the answer to all of these questions is probably yes. Everything we've worked on is this comparator circuit. And I think we have a reasonably good understanding of all the genetic handles to these circuits, right? I'm going to foreshadow a bit. Negative prediction error neurons are layer 2 neurons, superficial layer 2 neurons. The positive prediction error neurons are layer 3 neurons. These are different subtypes of SST interneurons, etc. And so we've identified all of these elements in the meantime. The way we've done this is always using mice and playing some form of a video game, right? The key experiment is this that we do is a mouse runs in a virtual environment, and then we can break this coupling between what it does and what it sees, and thus induce what we think is a negative prediction error, or we call it the stimulus, we call it visual motor mismatch. We halt the visual flow. And this, right, normally the mouse expects visual flow given it's running. We remove this, and this triggers what we think is a negative prediction error, the absence of an expected stimulus. We can control for the sensory aspects of this by repeating the animal. Now, notice the stimulus is identical. This is just a replay. We call this open loop or playback. And now the mouse just observes what it had self-generated previously, right? So, and this way we can control for what is sensory and what we think is actually a prediction error response. To give you an idea of what these responses look like, this is a visual response of layer 2-3 in mouse visual cortex. This is the response when the mouse self-generates the same stimulus. This is always larger. It's true in humans. It's true in mice. Nobody really understands this difference. I certainly don't. But what we then saw is if you look at these visual motor mismatch or feedback mismatches, as we called it here, you get a very strong response, and the passive control, where the mouse just sits there and observes the same visual stimulus come to a halt and then restart, does not trigger any measurable response, or at least didn't with the technology we had at the time. This was 15 years ago or so. So there's a big difference, right? And the thing we need to explain is the difference between the orange line when it's self-generated and the yellow line. And this obviously now we think is a prediction error. So this visual motor mismatch response is a good way to measure prediction errors. Right. So the question, and I'll briefly run you through about 10 years of work, just giving you how, images of how we did the experiment and what the conclusion is without going into the nitty-gritty circuit detail of how we did it. But one early objection that we had, or that was raised by the field, was that this might just be a surprise signal. How do you know it's a prediction error? And an experiment we did then is to show that it has receptive fields. So you can do this in this way. You can have a mouse run down a virtual corridor, and now instead of halting the stimulus only full field, we have these local patches that are indicated by these red arrows every once in a while. I hope you can see this. But the idea is relatively simple. We just, instead of halting the full thing, we just halt individual patches and can in this way show that these things are perceptive fields. So to a given neuron in part of visual cortex that codes for a specific part of visual space, only flow halts in that space actually lead to a response. Everything else doesn't. So it's definitely not some global surprise signal. Lending further credence to the idea that it's a prediction error type signal, right? To give you an idea of how we tackle the question of where does the top-down prediction come from? First of all, where in cortex, which cells does it target? The short answer is we can use, mouse tools like rabies tracing here. In the case of visual motor predictions, these we think come mainly from a motor area called anterior cingulate in the mouse. So in the mouse, this is a motor area, and neighboring the secondary motor cortex that form a very strong prediction to mouse visual cortex. So it's the region here in the back. And then we can use an experiment of the following variant. And again, I'm just going to explain what the idea of the experiment is without explaining all the data in detail. But the idea is that we have these signals. The top recording here is axons from ACC to V1. We can record from them. Now the question is, do they actually encode a prediction of visual flow given movement, right? And the things we need to disambiguate them from is a motor signal and a visual signal, right? And the way we did this is we trained mice in this video game where they had to navigate to the end of the tunnel, turn left, they had control over left and right of the tunnel. And then halfway through experiments, we switched left and right. We basically left, right, inverted them. And mice can relearn to do this. I mean, so can humans, obviously. If you flip your mouse upside down, you can still learn to use it. It takes a second. But then what we saw is if you were a visual signal, we would expect it to flip coding immediately. If you were a motor signal, we wouldn't expect it to flip coding. But what happened in reality is that as the mice learned to re-navigate, we saw this gradual flip of these signals that code for, as the mouse turns, now that as it learns that turning left also leads to left visual flow, we saw the coding space in these axons gradually change with learning. Based on this and a number of other arguments, we think that these signals actually encode a prediction of visual flow given movement. Right, so we've identified candidates for this. Essential to this whole setup then is that this is learned with experience. It's not hardwired from birth. We can do this stealing old ideas from Held and Hind, where we raise mice in complete absence of any visual input, and the only visual experience they get is in this virtual environment. So they're in complete darkness, except for 2 hours a day, they sit in this virtual environment. And then we have a mouse that has control over its environment, meaning it has relatively normal coupling. As it moves forward, the world moves back. And then have a non-coupled trained mouse that can do whatever it likes, but it just sees what the other mouse is doing. So it's not coupled. It never experiences coupling. And in this way, we could show that only the mice that have this coupled experience actually develop these visual motor mismatch signals. The mice that do not receive coupling don't have them. And about, interestingly for us, it took only about 20 minutes. So if you take this mouse now, it's almost an adult mouse, and its entire life has only gotten this non-coupled experience. About 20 minutes of experience in its life is enough to fully establish the system and then result in normal visual motor mismatch responses. And we can even show in this context, we have another paper that shows that for this to happen, plasticity in visual cortex or local NMDA receptors in visual cortex are actually necessary. So we think the plasticity that sets this up is in V1 in layer 2-3, and it's learned with experience. Right. I alluded to this already. We think we know the genetic identity of these cells using mouse genetic tools. We could actually identify what the marker genes of these neurons are, and then we're able to make viruses that we can use to express whatever our opsin of interest or our protein of interest in these cell types. The ADAMTS2, it's a marker gene. The gene doesn't really matter, and we don't understand why it's a marker gene. But and these names come from the Allen Institute. We didn't come up with these cluster names. All we did is identify that this set of cells is the negative prediction error neuron type. The BAS1A is the positive prediction error neuron type. These are deeper in what later in primates likely becomes layer 3. So the model we have now is layer 2, or the primate equivalent of layer 2. In mice is negative prediction errors, layer 3 is positive prediction errors. And then we identified the third type of cell in layer 2, 3 that we don't understand yet. So we don't know what they map onto. They don't have, they don't cleanly map onto our circuit hypothesis. Anyway, so we have identified these cell types, or these responses in mouse visual cortex in response to visual motor prediction errors. Now, obviously, most of the field hasn't used visual motor prediction errors, but other types we can show, right? These top-down predictions can be related to memory, what you remember seeing in a given location. We can do this in mice too, using an experiment that looks like this. So if the video stutters, this is just a mouse running down a corridor. And then every once in a while, it comes to a specific location where it knows or learns with experience that a specific visual stimulus is turned on. As mice do this, so this is now a schematic representation of that tunnel. It has these A and B stimuli, which are just relatively boring gray things. But the important thing is they only come on as the mouse gets to the specific location. So we know that the mouse cannot see the stimulus prior to the appearance of the stimulus. Nevertheless, we get cells that code for the, if you look at the black trace here, this is the activity of one neuron as a function of position in the tunnel. And you can see this black neuron starts increasing its activity prior to the appearance of the stimulus itself. And it's stimulus specific. It doesn't happen for locations where the mouse knows a different stimulus will appear. So we in essence can now decode what the mouse thinks it will see from activity in visual cortex about half a second before the stimulus actually appears, as well as we can decode it half a second later, what the mouse had actually seen. Anyway, so there are these spatially predictive signals, and then the experiment we did to show that there's prediction errors is now just emit one of these stimuli. This works as well, except it's experimentally very tedious because you need to train a mouse for 7 days in this environment, and then you have one emission or two events where something happens, where you can now emit this stimulus and you get a strong response. Anyway, I mean, experimentally, we don't use this very often because it's tedious, but in principle, we think there's also spatial prediction errors. There's other labs that have now reproduced these phenomena in similar settings. Now, this isn't our, I mean, we've worked on this, but I should highlight, obviously, most labs that have worked on predictive or circuits of predictive processing have used stimulus association, things like oddball stimuli, or associate a specific sound with a specific visual stimulus and then see if there's prediction error-like responses. We've done one such project as well, where we've associated sounds and visual stimuli, and then you get very similar sort of prediction error-like responses in layer 2-3 of visual cortex. And if you first, so the experiment here was pairing a specific sound with this given visual stimulus. Initially, you get in visual cortex, you get a response to the visual stimulus with experience in pairing. So the initial response is the purple one. You get the visual response. The visual stimulus starts at 1 second. The sound starts at 0. And then with experience, the blue line, you get the suppression of the visual response driven by the sound. And then at the end, if you show the wrong sound or no sound, you sort of unmask this suppression, which then ends up looking like a prediction error. Anyway, but I mean, this has been well described. The important thing is all of these things are much, much smaller than visual motor prediction errors. Right. So it works in other modalities in visual cortex. We also know it works in other cortical areas. This is a repeat of the first experiment I showed you now in auditory cortex with what we call audio motor coupling. The mouse learns, again, this is artificial, but it learns that the faster it runs, the higher the sound we artificially generate with the speaker, and then breaks in this coupling also trigger audio motor mismatch responses in auditory cortex. We've even used similar logic in motor cortex to see whether there's evidence of this. Here, it's slightly more complicated because it's not entirely clear what a prediction error in motor cortex would look like experimentally or how to trigger this. the experiment we used here is we trained mice again to run down those quarter, but now suddenly shifted the direction the mouse had to go. So we just rotated the world briefly, at unexpected moments by about 30 degrees, and measured how motor cortex responds to this perturbation. And we can compare that to how motor cortex responds when the mouse normally moves. And what we found, I hope you can see this on the right, it's hidden on my screen under the video. But under normal conditions, motor cortex, it scales with the vigor of the movement. So the faster the mouse turns, the stronger the response in motor cortex. This isn't a surprise. Surprising thing was that when we have these offsets, mice don't always correct. And now we can see what happens as a function of the correction of the mice. And what you see here at the bottom is the layer 2-3 neurons activate independently of whether the mouse corrects or not. So in these moments of sudden perturbation, there's this activation of motor cortex that's completely independent of whether the mouse actually turns. Sorry, the acceleration here is rotational acceleration. So 0, the mice just keeps running straight, and there's an activation that's as large as when it actually corrects for the full perturbation. Anyway, and we interpret this plus a series of other arguments to indicate that there are something like a visual motor prediction errors, or motor prediction errors in layer 2-3 of motor cortexes. Right. So does any of this translate to humans? I mean, obviously there's plenty of literature on oddball responses and all kinds of other responses in humans, but we specifically wanted to add one piece of evidence that there's also visual motor prediction errors in humans, which we had briefly worked on together with Klaus Einer, Stefan, and Jakob Heinz a few years ago, but never actually published this. And now I've done this with EEG and virtual reality recordings. And the answer is yes, you can replay this. So the experiment looks like this. It's just a human version of the mouse experiment where participants walk around in this virtual corridor and self-generate movements. And then all we do is we halt the visual flow at random times to see whether there's actually these visual motor prediction errors. And the answer is yes, this actually feels completely eerie. And funnily enough, this reminded me of old Helmholtz experiments that I'm sure you're familiar with the lesion to the external rectus and the patient that reported seeing the world fly to the right whenever he tried to move the eye to the right. What participants report here is that as soon as we halt the visual motor coupling, they report seeing the world fly forward. Right? It's not just halting the world, they really feel like the world is suddenly flying forward. And they often even trip slightly when this happens the first time. Anyway, the EEG responses are relatively strong and clean in over occipital cortex. On the left is this visual motor mismatch. On the right is the open loop replay. This is where participants just sit and watch the same replay. At 0 seconds is when we halt the flow, and at 0.5 seconds, the flow restarts, or we briefly need to teleport the subjects to where they actually are, right? If we halt the flow, and then half a second later, we need to restart it. And both lead to a response. This is something we never saw. The second response we don't see in mouse, at least not to our best experiments. But the first response is visual motor mismatch is exactly the same. It's attenuated just like in mice if participants only view this. Interestingly enough, the second response, the positive prediction error, if you will, seems to be much less influenced by whether it's active or passive. But the negative one certainly does. Right. So this leads me to the major crux. I think there's very good evidence that there are prediction error computations in layer 2-3 of cortex. I mean, this is what we've done over the last 10 years or so. Focus on trying to understand what the circuit elements are. We now have viruses that we can use to express in one type or another. A lot of this has been replicated now also by the Allen brain that reconstructed using EM data, these mouse visual cortex, and found cell types in the EM data that are consistent with exactly this circuit. Right. So far so good. The thing that has driven us to the point where we think this model is not a good model of cortex is the final one, the internal representation neurons. They're often speculated to be in layer 5. So did we, right? I mean, we always speculate that this must be layer 5. So we started doing a series of experiments to try to identify internal representation neurons, because these circuit diagrams makes very specific predictions about what should happen if you artificially activate internal representation neurons. You would expect the positive prediction error neuron to be inhibited and the negative prediction error neuron to be excited. And sorry, this is now an important point because this is different from the Rao and Ballard implementation. Their model flips this in this basically by having the internal representation neuron in a different layer of the hierarchy. The influence of the local ones flips. We can discuss the details of this later. But what I want to focus on, our evidence is consistent with neither. We can refute both models with what I'll show you in a moment. But it's important to keep this in mind. It depends on where you put the internal representation neurons locally. Our logic at the time for our proposal was that if you keep the internal representations neurons like this, you'll notice that they interact with the prediction errors like a prediction. So the sign of the influence, right? The prediction is inhibitory and positive prediction error neuron, so is the layer 5. The interpretation of this is that your local internal representation functions like a prediction for the local prediction error neurons. And this allows a system to work with one brain area only. You don't actually need a hierarchy. This works in a non-hierarchical way. You can connect these laterally instead of needing a hierarchy because you split this across different hierarchical levels. Right. So anyway, that's just to the detail. But Rao and Ballard makes the exact opposite influence. And if you Rao and Ballard flips both the influence. So the layer 2-5 or the internal representation, the prediction error influence is flipped with respect to this model, as is the prediction error to internal representation influence. Anyway, right, but we can now, because we have tools to express the channel rhodopsin in this, a candidate internal representation neuron, and then measure the influence in prediction error neurons. We can ask, is there evidence that layer 5 actually acts as internal representation? So the experiment looks as follows. We can express a channel rhodopsin variant called crimson in this case in our favorite layer 5 population. I'll show you 3 subpopulations of layer 5 and 6. And then measure the response of functionally identified neurons in layer 2-3. The experiment the data actually looks like this, where we record in some place in layer 2-3 and then can measure visual responses, can measure mismatch responses, and the response to the artificial stimulation of layer 5. We can then for each cell quantify how it responds to these different things and ask the following thing, right? Is a positive prediction error neuron, is that actually inhibited by layer 5 IT stimulation, right? We don't know which subtype of layer 5 could function as internal representation. That's why we just need to screen all of them and to see which one is consistent, right? If we do this for layer 5 IT, which was our prime candidate at the time, we do find something that's absolutely non-trivial in that positive prediction error neurons are excited by layer 5 IT stimulation and negative prediction error neurons are inhibited very strongly by layer 5 IT stimulation. This is the exact opposite of what Tom and I had proposed at the time. It is consistent with Rao and Ballard, right? So this is exactly what Rao and Ballard would predict. It's not what we predicted, right? So if we repeat this experiment now for layer 5 ET neurons, this PESF2 is just the mouse line we use to target them. It looks exactly the same. The same is even true for layer 6 neurons. So NTSR1 is layer 6 and deep layer 5 neurons that we can target. In all of these cases, we get excitation of positive prediction errors and inhibition of negative prediction error neurons, which is inconsistent with our model, consistent with Rao and Ballard. Right? So far, in principle, one could exclude our version and still uphold Rao and Ballard. And then what we did is so far, right? Sorry, I'll skip this. Well, we can do the experiment the other way around. We can stimulate now layer 2 subtypes, positive prediction error neurons, and see how it influences these layer 5 subtypes or negative prediction error neurons, and see how this influences these subtypes. And again, our model would predict that positive prediction error neurons excite internal representation neurons, and negative prediction error neurons inhibit. And this is exactly what we found. Right? This is now inconsistent with Rao and Ballard, but consistent with our model, right, for an internal representation neuron. So our data rule out, as far as we're concerned, both models. I'll give you an upshot. So what we think is going on is that, so the other thing that this is consistent with is that layer 5 acts like a bottom-up input to layer 2-3. Right? And I'll give you one more piece of evidence for this. Something we had noticed through other research, and not just us, people in the field had noticed that you can actually decouple layer 2, 3, and layer 5. And we stumbled upon this by giving mice the antipsychotic clozapine. So if you look at a brain-wide recording or a layer 2, 3 recording, there's basically no effect of clozapine on layer 2, 3. But there's a humongous effect of clozapine on layer 5. So I left this before clozapine, right this after, and you can see there's a little bit of an effect. I hope this translates on Zoom, but there's not much happening if you look at layer 2, 3 or most of the brain. But if you look at the right layer 5 IT subtype, you get a very strong effect of the drug, which is peculiar, right? How can what we with other work have now shown that what an antipsychotic does is decorrelate layer 5. But how can something decorrelate layer 5 without decorrelating layer 2, 3? Very similar things. We weren't the only ones to find this. Anesthetics do this as well. They cause sort of cortex-wide synchrony of layer 5, but leave layer 2, 3 relatively untouched. Ketamine at sub-anesthetic doses does the same thing. In the meantime, we could show that the activity patterns in intragranular layers, deep layer 5 and 6, are almost always orthogonal to layer 2, 3, not just in with antipsychotics, anesthetics, or under ketamine, that they almost always don't correlate all that well. It's almost like they do their independent thing, right? So with the way I used to think about cortical function is that the dominant axis of communication is vertical, meaning if you ask what is the dominant influence on a layer 5 cell, I would have said until recently it's superficial layer 2, 3 communicating with it. Ever since seeing these results, I think there's much better evidence that the dominant communication is lateral, meaning that a given layer 5 cell receives much more of its driving input laterally, which obviously is inconsistent with the idea that internal representation neurons are in layer 5 and prediction error neurons are in layer 2, 3. So we think we can decouple them. So what does that mean? What we think is going on is that layer 5 actually is the bottom-up input to the layer 2, 3 prediction error circuit. So what layer 2, 3 is doing is not predicting bottom-up input, but making a prediction in latent space. It's trying to predict the activity of layer 5. And one rather pedestrian experiment we can do to address this is to artificially just ask, can we induce prediction errors in layer 2, 3 by artificially coupling layer 5 activity to locomotion or something else? Well, we chose locomotion here because that we think again is the most dominant form of prediction that we have for movement in general. Right. So if we normal visual motor coupling, I showed you this already. If you break this coupling, normally locomotion and visual flow are coupled. You break the visual flow, you get a strong response to the decoupling. We can now artificially couple locomotion speed to layer 5 activation. So the mouse is in complete darkness. We're in visual cortex. We just activate layer 5 artificially as a function of locomotion speed. And without any training, we get the same neurons that are activated. So almost equal strength. And we can even show it's the same neurons that are activated by visual motor mismatch and this artificial decoupling of layer 5 stimulation with locomotion. Right. So obviously none of this is conclusive evidence, but it definitely seems to point in the direction that, the cortex might be better understood in terms of a JEPA than our predictive processing circuit. If you're not familiar with a JEPA, it's this idea that it's from Jan Lecan, and the idea is that you have two networks in parallel that receive the same input, where one learns to predict the other, and this in essence prevents representational collapse in self-supervised learning. I'm not entirely sure it's fully understood why it works, but it seems to work quite well. And work quite well as in machine learning, work quite well. There's some parallels to cortical architecture. You have two input pathways to deep layers and superficial layers directly. We now know that they can be decoupled under certain circumstances and may often be actively decoupled. And it looks like layer 2, 3 computes a prediction error with respect to layer 5 and not bottom-up input. Right? So our working model at the moment is that there is a predictive processing like architecture that works exclusively in superficial layers that whose job it is to predict activity in deep layers. What the architecture or the cell types are in deep layers, we don't know yet. And they communicate importantly in a non-hierarchical fashion, right? One of the big problems that we really haven't learned to address, I mean, with the exception maybe of the work of Rafael Bogac, how to get these things working on arbitrary graphs, right? Cortex is not a hierarchical network. It's an arbitrary graph, and we need to find ways to solve this problem of understanding how these algorithms might be implemented on arbitrary graphs, networks like Cortex. Very good. I think that's all I had prepared as a brief overview, and meant to sort of guide to where I think the open questions are, right? The things that we really haven't understood is in any meaningful ways, how is precision guided in this predictive processing network that may be implemented in layer 2, 3? What does the interaction between layer 2, 3 and layer 5 look like? Because as far as we can tell, it doesn't look like layer 5 functions as an internal representation for the prediction error circuit in layer 2, 3. How do we get this to work on arbitrary graphs? Yeah, I think those are at least experimentally, the big unanswered questions.
Thank you, Georg. What a ride. So the custom is that we briefly unmute our microphones and give you a round of applause. Thank you.
Thank you very much.
So now there's time for some discussion from the group. I don't know, Carl, if you prefer summarizing in the end or keeping it more informal this time.
Both. Yeah.
Both. Good. And I mean, I often start out just to frame the discussion, maybe with also what would be the most useful to focus on. Partly to refrain from just asking about anything I found vaguely interesting or random thoughts I thought about while you talked, because I have a long list of those. So maybe that's a good place to start for now as well.
Please.
Yeah. So what do you think, Georg, would be the most, and maybe also the rest of the group, the best place to start? I mean, just thoughts on the open questions, or
completely up to you, whatever is most useful.
Okay. What do you think, Carl and Thomas?
Well, so I can certainly highlight some of the themes I thought were really interesting. I mean, everything was very, very interesting there, and it's a sort of really impressive array of work. I have to confess, I'm struggling to keep all of the ideas and themes in my head, given how much there was. I think some of the key themes that I thought were really interesting were sort of number one was thinking about the specific relationship with the anatomy and the laminar anatomy. And I suppose that is one of the key scientific questions, isn't it, of exactly how one pins down a sort of computational, theoretically derived procedure for inference to specific anatomical circuits. So I guess that's one of the key themes that's sort of come through a lot of that research. The second theme that I thought was quite interesting was the relationship with action and motoric components. And you motivated that really well, sort of thinking about forward inverse models and all that sort of thing very early on. I thought that was quite an interesting theme to pick out in that a lot of the very early work, as you know, on predictive coding, the Rao and Balil type stuff, also a lot of the sort of thing that people like Rafael Bogac's work on often is much more focused on the perceptual sensory classification side of things. So it's really interesting actually that a lot of your choice of focus was much more on the action and the consequences of what I do. And I thought that the third theme that perhaps wasn't quite as explicit, but I thought was interesting, is actually the physiology of it, in that the idea that there may be separate layers representing both positive and negative prediction errors. Whereas often in the sorts of diagrams we might write, theoretically, we might sort of assume a common population that is somehow within that population representing both. And you could make a whole series of arguments as to how that's done, whether sort of, as you've intimated, it's separate populations doing the positive and negative components. Or you could assume a sort of log code where you sort of have to take the logarithm of the firing rate in order to get both positive and negative values. or if it's just in the sort of overall effective inhibition versus excitation. So there's lots of interesting elements to that as well. So for me, those were the sort of three key themes that I thought would be really interesting to discuss further. So sort of the anatomy, the effect of action, and the relevance of prediction based upon what I'm going to do. And the third bit being exactly what we think these different populations are doing, how we're actually encoding these things physiologically. Do those sort of fit with what you think of as being some of the key controversies or open questions, or have I slightly missed the point there?
No, I think that fits quite well. I mean, to most of the physiologists, it is indeed the question of, how do these elements, how are they implemented? Right? And the first and the last of your points actually merge almost into one, right? What is the anatomy? Is it a divisive computation? Is it a logarithmic computation, or is it just subtractive?
Yeah.
Which then will have different answers in terms of circuit elements that are involved.
And it sounds like the answer you'd give is that it is sort of more directly subtractive if there are sort of clearly identifiable neurons representing positive or negative components. Well, perhaps I can ask that. So, what sort of tells you that it's a positive versus a negative prediction error?
Yes, this is in essence is interpretation, right? When we remove when we don't present an expected stimulus, in this case, visual flow is, I mean, we think of, especially in mouse visual cortex, the coding is probably in visual flow speed. And then removing expected visual flow given movement, just by definition, is then a negative prediction error. Or I mean, one can flip it around depending on what one considers. That we speak now of teaching signals. This is, in sensory space, that will be the signal from the world and the prediction, and those two are compared. And one can flip it around. Our model now is that all areas in this non-hierarchical graph of brain areas exchange both predictions and prediction errors bidirectionally, and they can both act as a teaching signal for the other. But in visual cortex, I think we do have evidence that there is simply just a subtraction going on. We can definitely identify neurons that respond to what we think of as a negative prediction error in visual motor space, or a negative prediction error in spatial visual space. So we find neurons that code for these things simply with an increase in firing rate. Right? So it's genetically identifiable neurons that have this opposing influence. Right? What I didn't show you, we've also done intracellular recordings from these neurons, and we know that they receive opposing influence neuron by neuron. So you take a given neuron, if that neuron receives motor-related excitation, sorry, I should be careful. We don't know if, so I'll speak of excitation right now and clarify in a second what I mean. There's an experimental caveat to this. But if a neuron gets motor-related excitation, we know that same neuron will get more visually driven inhibition and vice versa. And so exactly what you would expect from two populations, one that encodes positive prediction error and negative prediction error. They need this opposing influence. Sorry, and with respect to excitation, we can only do current clamp and not voltage clamp. So we don't know whether it's a hyperpolarization versus depolarization. We don't know whether the currents are inhibitory or excitatory. But at least it's consistent with this interpretation that they receive opposing influence, neuron by neuron, right?
So there's a clear sort of metric in one direction or the other, but as you say, you could flip the direction of that.
Correct. The interpretation of which is which, but I think there's very, very good evidence that it's just subtractive. It's the simplest implementation, seems to be the one that, at least in the mouse, seems to have been chosen, in the sense of you can compute the prediction error simply by subtracting two signals, and we have all the cell types necessary. It doesn't require any population code. Obviously, it might be doing a combination of these things, but there's definitely evidence of a subtractive computation of prediction errors.
Okay. And just because you mentioned it there, sorry, I'll give way to somebody else in a moment. But the teaching signal you were talking about, what sort of teaching are we talking about? Are we talking sort of long-term plasticity effects, or are we talking a sort of shorter-term modulation?
Right. So just for nomenclature, yeah, this is a tricky one. We've been struggling with how to talk about this. But the idea is the following: is that if you have a prediction about what the visual consequences of a movement are, that prediction needs to be learned, and the teacher in that sense is the world. Right. So the normal way in predictive processing of speaking of the teaching signal is a sensory signal. Right. So in visual cortex, up to recently, we would have spoken of a sensory signal being compared against a prediction. But this terminology doesn't work if you're in motor cortex, or if you are in, how do you talk about this in communication between auditory and visual cortex? Right. There's one of the largest inputs to visual cortex in the mouse is auditory cortex, and in the primate actually. And these are likely cross-modal associations, meaning auditory cortex sends a prediction of a visual response given the auditory scene or the auditory stimulus to visual cortex. But that, how do you talk about this in the context? Both are sensory signals, the auditory and the visual signals, right? So anyway, so we've opted to talk about this simply as a teaching signal in the sense that this is the signal that trains the prediction. And depending on, and now this becomes it makes lots of assumptions about which one is the plastic one, which was in this teaching signal, but this is simply semantic. We define the signal that trains the prediction as a teaching signal, which in visual cortex and visual motor associations is just a visual signal.
So it's the signal against which a prediction error is calculated.
Yes, exactly. It's the signal against which the prediction error is calculated and against which you learn, right? If you want to minimize the prediction errors, then and the predictions are the thing that are plastic.
Yes.
Presumably in reality, I assume both are to some extent plastic, but abstractly we can now speak of you train the prediction given a certain teaching.
Yeah. I mean, I have a lot more questions, but I'll let somebody else have a go first.
It was a minor one and perhaps an ignorant question too, but it's just given what you were just discussing before with how you know that they're positive or negative populations. So just open question, are we given is it possible to distinguish them from, for example, encoding something that would be a consequence of a positive or negative prediction error? So I'm thinking of something like the precision of the prediction. It would supposedly change differently depending on the type of prediction error, maybe.
No, absolutely. So we don't know. So if you just observe the responses, you have no idea whether it's a primary response to the prediction. A good example is a positive prediction error. Right? If you sit somewhere, let's take an experimentalist that puts a monkey or a cat in a chair and shows them some visual stimulus. The response to that, we typically talk of that response as a visual response. At the same time, you can think of this as a positive prediction error, right? You have no expectation of what's going to come on the screen, and suddenly there's a stimulus. So now that response can either be a sensory response. I mean, this is terminology and semantics, a positive prediction error, or even in predictive processing, there's now a confound because both positive prediction error neurons as well as internal representation neurons are expected to increase their activity in response to an external stimulus. So simply from the response alone, you can't tell, right? It doesn't even help semantically distinguish. The reason we actually focused initially on negative prediction errors is because I didn't want to get into this discussion with the rest of the visual neuroscience physiology community that has described for a century almost described these responses as sensory response. Right, so the negative prediction error was the response that was very hard to explain in a classic view of how visual cortex works. But the response alone, sorry to answer your question, doesn't actually allow you to distinguish, right? Because you need to. That's why I mean everything I've shown you doesn't, beyond this shadow of a doubt, prove that these are actually a subtractive computation, of course. But everything I think that's experimentally feasible or has been until now, we've tried. And everything is consistent with a subtractive computation of two signals, where one is a sensory or teaching signal and the other is a prediction.
Yeah, and you said it's the simplest explanation for the data right?
Yeah, and in my mind, it's also the simplest explanation. Obviously, there's always others. I mean, there's explanations that most of my discussions in scientific discourse actually revolve around whether this is even predictive processing, right?
Mm.
Or whether it's a combination of a stimulus-specific running modulation gain. But you can always, it's a bit like discussing whether the world is heliocentric or whether there's
Mm.
epicycles. It's both models, if you allow for enough complications, are perfectly consistent always with the data. And I think we're in a similar context. And most of the discussion outside of, sorry, in circuit neuroscience is versus, is it even a prediction error? And yeah, the short answer is we can't prove that. But at least everything we can do is consistent with the subtractive computation. Sorry, I'm repeating myself.
Thank you. I have another brief one, and then I'll give it back to you, Thomas, or Carl. About prediction errors in motor cortex. So again, this might be I'm usually at the computational level, so I'm not so physiologically expert at all. But of course, something like a proprioceptive prediction error or a conclusion that I did something other than I thought I would do. I'm not sure if that necessarily would be located in the motor cortex, but that's at least my intuition. That seems different from the stimulus or the sensations I got are different from what I thought they would be given what I did. Yeah.
Yeah. So I mean, to be honest, I have a hard time thinking about motor prediction errors in this context as well. So I mean, a somatomotor prediction error is likely computed in somatosensory areas. Sorry, it's my speculation, right? It's because motor cortex generates a prediction of what you should feel, and then you compare that against what you actually feel in somatosensory areas of the cortex. Now, if you turn this around, right, what motor cortex should receive is a prediction of what it should be doing as a function of somatosensory activity, right? So given somatosensory or given visual activity, this is a prediction of what you should be doing, right? So you're computing now a prediction error against the incoming information from other brain areas. And here I think my intuition just breaks down a bit as to how I would exactly expect this to look, or how to do an experiment that neatly demonstrates that this is a prediction error. Even in the work I showed you, right? What we read, the only thing we could come up with at the time is argue that the response doesn't look like it, whatever's going on, it's a response that doesn't seem to depend on what the mouse actually ends up executing. Because it's like a different code from the motor code you can recover if you just try to correlate the activity with what the mouse is doing. Because now in this context, you have no idea when prediction errors happen, right? Because then if you average over, it's a bit like in visual cortex. If you were to just average over visual stimuli, what you would be left with is visual responses, which is what obviously everybody has been describing and measuring in absence of knowing what the predictions are. And in motor cortex, we're in somewhat of a similar situation. My interpretation and my limited understanding of this, that we just don't know enough about what the predictions are it receives to compute prediction errors against. Basically, what is the incoming prediction? What is even the teaching signal in motor cortex?
Yeah.
And this is just in my mind. Sorry, obviously conceptually I understand, but how to turn that into testable predictions that I can test in a mouse experiment, right? And then identify the responses, like abstractly. I understand there are obviously beautiful ideas about how this could work, but we haven't been able to turn those into, when I say we here, I mean the field. I don't think anyone's got a good idea of how to turn that into a physiologically testable prediction as to, in a certain testable way, not just bulk responses of the entire thing.
What do you think, Thomas? Could you expect either if your planning changes, so you end up realizing you will be doing something else than you thought you would be doing, or maybe more back in time, if you are uncertain about which actions you used or you executed just before or are executing now. That's at least my own intuitions that as a behavioral
So
modeler.
I didn't quite understand the question. So
No,
I appreciate those are two different sources of uncertainty, or I guess predictions that one could make. Are you suggesting that those would be the signals to that one might be able to use in motor cortex, or
Maybe it's a bad suggestion, but just yeah, like what would be the prediction errors one would have in the motor cortex? I guess it's the open question, right?
I see what you're asking. It's not suggesting it's a silly question at all.
Okay.
The so there is some theoretical work on this from many years ago with I think people like Rick Adams and Stuart Shipp and Carl was obviously involved in much of this as well, asking questions about laminar structures in motor cortex and how they appear to differ from different parts of the brain. And I'm summarizing this despite not being involved in it and there being someone on those papers in the discussion. So Carl may wish to take over. But my understanding is that the argument is around the number of cells in layer 4 in motor cortex in many species compared to other parts of the brain, and that layer 4 often being in receipt of these sorts of error signals and sensory correction to predictions is in many ways less relevant for motor cortex, because if you can make good predictions that are then fulfilled, sort of predictions of the proprioceptive consequences of action that are fulfilled by spinal cord reflexes, then there effectively is no prediction error at that level that gets transmitted to motor cortex. So it effectively becomes the sort of automatic resolution of those errors means that one doesn't need quite the same degree of prediction error making it to that stage. Now, clearly, there will be elements of proprioceptive prediction error in some circumstances. If there's a barrier you come across, there may be somatosensory changes you weren't expecting as a consequence of that, and you could imagine some of those coming to other neighbouring areas of cortex. Carl, did you want to add anything more to that, given your more direct involvement in that research?
Yeah, you caught me off guard. I'm not used to speaking halfway through. Yeah, no, that was I think a very nice summary. Perhaps Thomas, I don't want to disrupt your flow. You clearly want to walk through these things, but I'll just give you my little perspective here. It is clear, and I think we should remember this, given the agenda of why we're sitting here at the moment, which is to try and provide a framework to unpack where the big questions lie for the populace. And hopefully neuron in the fullness of time, although I'm disenchanted with trying to get stuff into neuron. In that spirit, it is clear to me that the kind of predictive processing, I did notice, George, and forgive me, I'm going to call you George because I can't do the Danish thing. It's clear to me you've gone beyond predictive coding. You're in the world, generic world of inactive predictive processing, which is I think a more intriguing but more encompassing world. So the very fact that you started with people like Danny Walpert and much of the experimental paradigm is about sensory motor integration, and you're using the intentions to move and possibly realize movement as a source of prediction. In my world, this is active inference, this is active sensing, active vision. It's nothing to do with predictive coding, read as even up until I think Rajesh Raya's work, bearing in mind that predictive coding had an earlier history. And just to exemplify that, it is only now, literally in the past 8 months, that Rajesh Rao has now turned his attention to action and motor activity, and he's actually introduced an active predictive coding. I don't know if you've read he's got a couple of papers on that. That tells you something. The theoretical community has not been thinking about action in the context of predictive coding until very, very recently. We have been doing this through the lens of active inference, and what Thomas just summarized was, I think, a nice take on that. So, from that point of view, the predictions now become the intended movements, and they arise from autonomous dynamics. So they're not a response to any stimulus. they are the kind of things that I do in this particular context. So you have central pattern generators that are driven technically under sort of a generative model that would produce particular trajectories and movements. So your prediction is generated in a top-down way, and that prediction is exactly the motor command, if you like, but it's only unpacked as a motor command in the sense it just provides the set point for much lower mechanisms that could be at the level of the pontine nuclei right down in the spinal cord that engage classical reflex arcs. So the prediction error is literally encoded by alpha motor neurons that drive the muscles. So the prediction error never needs to get back to the CNS because its job is to drive the muscles. So you tell your motor plant what the particular pose or trajectory that you anticipate or predict. You then elaborate a proprioceptive prediction error at the level of the spinal cord. That prediction error then drives via the neuromuscular junctions contraction until the stretch receptors and proprioception then match your top-down prediction. And this is just a restatement of in the 1980s, one side of the great debate prior to Daniel Walpert and Chris Miles' forward inverse formulation, called an equilibrium point hypothesis. So this is how you would make predictive coding active. You just equip it with reflexes. And on that reading, you would not need BET cells. You would not need any afferents to or prediction error signals at the level of motor cortex, because the motor cortex is generating the prediction that is then realized, and that's why the animal moves. So I think that's what I think Thomas was referring to, which is a kind of strange inversion. So the prediction errors now are not found in the cortex; they're found in the spinal cord because they are the things that drive the movement. And this is just a retelling of a story which was endemic in the 1980s. Well, even before that, I would imagine, and predates this forward inverse model. And in my world, the forward inverse model is now anachronistic. There's no evidence for it, and it just doesn't work mathematically because the forward inverse model doesn't address the question, where do my intended movements come from? It assumes magically somehow that this is the movement. And then all the forward inverse model does is try to sort of finesse and put forward as a particular way of accounting for axonal conduction delays and the like. But you just don't need that, if you just go right back to the equilibrium point hypothesis. So from the point of view of framing issues that need to be foregrounded, should we commit to cheekily sort of framing the issues for the populace? I think distinguishing between inactive applications of predictive coding from vanilla predictive coding of the sort that visual neuroscientists had been dealing with, and of course people like Rajesh Ray were talking and Dana Ballard and all of that work, including even nowadays people like Lars Muckley in sort of visual at a systems level. They're not considering the really important aspects of active vision, whereas you are. So I think that's a big divide which needs to be highlighted, because in virtue of this conversation, one's bringing along a lot of extra issues and quite deep sort of both theoretical but also empirical issues to the table as soon as you put motor behavior into the mix. Those are my thoughts. I've overspoken, so I'll shut up. And Thomas, I'll pass back to you, or ask George if he wants to. So I noticed, George, at times you were talking about predictive processing, and you very seldom mentioned predictive coding, and I thought that was really interesting. Do you know where predictive processing came from?
Well, my first encounter was with Andy Clark.
Yeah, yeah, it's from philosophy. So yeah, and I was behind the scenes when he was writing that his whatever next seminal paper, and the one of the reasons that he chose predictive coding as opposed to, sorry, predictive processing over predictive coding was exactly for the issue that you brought to the table and we've been discussing about. He didn't want to limit it to simply sense making. He
Yeah.
wanted to say that there's something deeper going on here that speaks to his inactivist embodied agenda, which of course he'd spent a decade promoting. So he invented the phrase predictive processing simply to accommodate action and motor behavior and choices and decision making. And I was quite cross with him at the time in a tongue-in-cheek way because he should have been calling it the free energy principle or active inference. And the reason he didn't was he didn't want to burden himself with all the theoretical commitments of the free energy principle and active inference. So he opted for the predictive process as a middle ground, which was, if you like, neutral in terms of its commitments to the physicists' take on these things, but didn't wed itself to predictive coding read as a visual physiologist, neurophysiologist would read it. So I was entertained by the fact you prefer predictive processing to predictive coding. I think that's something that has to be said out loud, because when we wrote that neuron paper, I thought they just wanted predictive coding. We tried to slip in action at the end, but the focus was very much on predictive coding. But clearly, in virtue of the fact that you've spent your entire career, and presumably a lot of people, a lot of your team has, and other people around the world, clearly the notion of predictive coding has now transcended its original limitations and now explicitly includes action and behavior.
To a large extent, I agree. I think many people still, so what I've noticed is people start using it synonymously. I mean, unfortunately, when Tom Maschert-Flögler and I wrote a review that was meant for systems neuroscientists in 2018, where we made this proposal for the circuit, we elaborated on this, and for the reasons that exactly the reasons you specified, we chose to call it predictive processing and try to distinguish it from predictive coding. Sort of the idea that there's a hierarchy. I often now speak of a hierarchical implementation that is a sensory processing stream, which for me just never mapped onto what cortex does, because for the very nature of it not being hierarchical, right? If you will, right, coming back to the motor cortex argument. I mean, the active inference, your active inference idea, when I first saw this, maybe, I don't know, 10, 15 years ago, I was instantly enamored with it for the very reason that, so I'll share just so I can quickly explain what I mean. We have, right, if one were now in motor cortex and you have a circuit like, so I'm just trying to find one of the circuit diagrams. Yeah, that's this one. But the way I read it at the time, and I assume it's probably what you also mean, is that the absence of layer 4 means that one way to think about the activity in motor cortex is a teaching signal that's clamped at 0. It doesn't get anything. So any prediction automatically in layer 2, 3 of motor cortex now produces a prediction error, but is the active inference interpretation. Now, the classic motor cortex codes for movement interpretation is unfortunately physiologically almost identical. You will find neurons that look like they code for movement. Right. So the question we've been asking ourselves for a long time, I don't think I have a good answer, right? Is that how can we design an experiment that would distinguish between these two possibilities? And for this, I always felt like we need to understand what are the incoming signals, assuming that they are predictions. What do these even code for, right? The motor cortex gets a ton of input, axonal input from regions like somatosensory cortex, even visual cortex, projected motor cortex. So what do these signals send? What is the information content? Is this really a prediction, right? Is this what drives the intention to move? I don't know, seeing a coffee cup and now that generates the prediction of standing up and grabbing or reaching for it or whatever, the movement necessary to get the cup. Is that the prediction that is sent? And is that compared against the null prediction? I mean, The motor cortex does typically have a little bit of layer 4, but obviously much less than the rest of cortex. Maybe a vestigial form of layer 4, not entirely clear, as far as I understand. But right, how can we use this idea? I mean, basically, the active inference, your active inference work was the inspiration for our motor cortex paper, right? To then ask, can we find any evidence of something like a prediction error? I don't think we did a great job at the time. We did not. Also, technology now would be better to actually address this. So, no, I agree with you. I think this is a message that I think one should very strongly drive home. And just to maybe, because you you mentioned that Andy didn't use the terms active inference or a free energy principle to talk about this. I always felt like predictive processing was a circuit proposal for how these theoretical ideas could be implemented in the brain. I mean, that's why I also find your idea, I wasn't familiar with this idea prior to reading your review of mortal computations. I think this is, that all feeds together into this, the idea is beautiful conceptually, but how on earth is it implemented in this wet system we call brain? Because it's likely, under diverse constraints, likely not going to find the optimal solution, and it's going to find some intermediate solution that beautifully is captured by the idea of mortal computations, I find.
Do you want to ask some of your other questions, Thomas?
Sure. So I guess why don't we stick with the theme we're on at the moment then, I guess, which is the motor and consequences of action side of things. So the first question is, can you just remind me when you do the rabies tracing work, that's sort of anatomically sort of lamina selective, but not cell type selective. Is that right? Or how selective are you in terms of the postsynaptic neuron that you're then looking for
yeah, no, that always requires, or at the time when we did these experiments, always require the mouse Cree line, which is a mouse line that labels a genetic subset of cells, which tend to be, the tools that we have available are either layer specific excitatory neurons or inhibitory neurons. And this is simply for historic reasons. Those are the tools that were meaning we could trace from all SST neurons, but not layer specific, or all PV neurons, or all excitatory neurons, but there we typically have layer specificity.
Fine. So the results you were showing us in terms of the way the motor cortex, or the sort of presynaptic neurons that we were seeing in the motor cortex, they are presynaptic to which population specifically?
So the axons, sorry, I need to be careful that I remember correctly. So the short answer is the primary source of the projection from motor cortex is layer 5.
Okay.
The target is almost everything but layer 5, IT neurons. So this isn't all or nothing. We always find synapses onto these cells, no matter what the cell type is, it will always target. There are preferential connections, and if we then look at the functional influence, which is the jargon we use to asking the question, what happens if we artificially activate all of these axons? So we can inject the general rhodopsin in motor cortex, for example, and then look at the axons in visual cortex and artificially activate all of them, and then quantify how do the different cells respond. The idea is that by this, we measure the direct synaptic connections, the indirect ones, the tertiary ones, all basically the entire influence spectrum. It doesn't need to be monosynaptic. It very likely isn't. But then measure how does it influence the activity of a cell. Obviously, this has a ton of caveats because we're not activating it in any natural activity pattern. We're just acutely activating everything. And then you see interesting biases emerge, namely that the cells that are negative prediction error neurons tend to be preferentially excited by this projection, and the positive prediction error neurons, the more sensory responsive ones, tend to be inhibited by this projection, etc. So we get the right biases that you would expect based on the model. But this isn't apparent from the from the rabies tracing anatomy that we could do at the time. We also, when we did these experiments, we didn't have yet the genetic tools to target only the positive and negative prediction error.
Okay. And the layer 5 side of things is quite interesting as well. In that, do correct me if I'm wrong, but my understanding was that a large proportion of the layer 5 targets are actually subcortical, sort of second order thalamus, basal ganglia, spinal cord if you're in the motor cortex, rather than, obviously clearly does have influence on other cortical areas. And that strikes me as being very relevant to the story of sort of action outcomes, consequences of what I'm doing, if I'm also then involving a number of the structures that we tend to think of as involved in planning and action selection subcortically. Is that something that these tools are useful for, or is there any work that you guys have done or noticed any findings in those other areas? Or is cortex being specifically the focus here?
For us, the focus was always the cortical algorithm. There's quite a substantial amount of work looking at cortical thalamic loops and their involvement. Sonja Hoffer has very nice work showing that visual motor prediction errors, for example, also arrive by secondary thalamus to visual cortex. I think both of our interpretation now is that it's likely a way to distribute. So the thalamocortical loops are used to some extent to distribute the prediction errors to more cortical areas, or to actually relay it via thalamus. But this entire, as far as I'm concerned, and I assume there are many people who would disagree with this statement, is that we know far too little about thalamocortical loops to draw any conclusions about how this would work. It's definitely feasible. Somebody should, and people are, right? There's lots of very, very good labs that are pursuing these questions, but I fear we will only have answers in a few years to ask exactly these questions about, how does this integrate with the thalamocortical loops or even corticostriatal loops?
And part of the reason I'm asking that is actually to try and get at that question of what sort of inputs are going to be relevant to the motor cortex to even decide what it is I might want to do. Certainly some of the work we've put together has suggested one could interpret some of the cortico-basal ganglia thalamic loops as being sort of evaluation of a selection of alternative policies I might have proactively based upon information represented in the cortex. There's ways of calculating the relative goodness of different paths into the future. And I wonder to what extent that might be an important ingredient in that focus. So I suppose one question is, how do I predict what's going to happen as a consequence of my actions? The other is, how do I use what those predictions are as a way of actually determining which action I want to take? And I suppose it depends which question you focus on, but that's perhaps another direction that might be relevant in terms of thinking about open questions for the field.
No, I see. I think it's an excellent question. There's one big caveat I think that you may or may not be aware of when it comes to this experimentally, just a rather technical, silly caveat. The mouse doesn't actually use its cortex. So 99.9% of mouse behavior does not. You can take out all of cortex in a mouse, and I will challenge you. I give you a mouse without cortex and one with cortex. You will not be able to tell me which is which. Right? This is the polar opposite of humans, where humans are the only animal where a lesion to motor cortex will prevent you from using that part of your body for the rest of your life. Right? The impairment is lifelong. Not even non-human primates relearn within a few months to use the affected part of the body. Humans do not. Obviously, we recover after a stroke and things get better, but that is almost always simply due to the surrounding tissue, with a bit of plasticity relearning. But the core region that you've lost, you never, right? You're blind for the rest of your life if you have a large enough stroke in visual cortex. Mouse behavior does not depend on cortex in almost every single circumstance that we can test experimentally. Obviously, there are things we can do, but you really, basically the answer is a rat or a mouse probably uses its cortex once in life when something very, very unexpected happens. Cortex functions to provide a behavioral alternative that can then be executed. And without a cortex, the rodents just don't know what to do in these situations. Adam Camp, who recently died, unfortunately, had this beautiful series of experiments where he trained rats to run across horizontal ladders, which are just these sticks that the rats traverse and run back and forth. And they can learn, it's not something they do normally, but they can learn to do this without motor cortex. It doesn't matter. They train it. You don't actually notice any difference in the rats doing this. The only time he saw a difference is when after training, he would let one of these rungs suddenly rotate. So as soon as the rat steps on it, the rung rotates and the rat basically slips. So he runs across, steps on this, and slips. And here's this is all published, and there's this beautiful series of videos where he just shows 6 or so of these rats without motor cortex. They slip and they just freeze. They stop moving. So when they encounter something unexpected, they stop moving. A rat with motor cortex will do something else. Will jump away, will sniff, will escape, will panic, just do something else. Whereas the one with motor cortex lesion just freezes and doesn't know what to do. Obviously, it takes 2 or 3 seconds. This isn't long, right? But within 2 or 3 seconds, it then does something else and recovers. But that 2-second advantage is likely why a rodent has a cortex, simply because that's that one time in life where that reflex saves it from being eaten by the owl is good enough, is justification enough evolutionarily to have a cortex. But that's likely all it does in mouse behavior. So meaning, right, there's a very, very profound problem when now thinking about behavior in mice and try to testing many of these ideas with circuit approaches, is that motor cortex isn't used in most. I mean, that's why this paradigm that I showed you where we have this sudden offset, that's one of the few instances where we know motor cortex is actually relevant to correct. So if we silence motor cortex just at that moment where there's the correction, mice tend to fail to correct for the offset. It's those few moments where motor cortex is necessary, unless you can actually sort of, then design a paradigm that focuses just on these movements, the corrected movements, the unexpected errors. It becomes very difficult to ask these types of questions in the mouse, using mouseology, if you will. Right. Anyway, sorry, I think that's technically what's hindering the field, is that, well, mice just don't use this structure. This I think is the same problem in psychiatry, right? That's why most behavioral assays of psychiatric drugs in rodents are just useless. Most of the behavior doesn't depend on cortex, so you can't screen for antipsychotic efficacy in mice using behavior. Sorry, I'll end my rant there.
No, really interesting. And I suppose it speaks in many ways to what Cole was saying about the use of sort of central pattern generators and reflexes at a spinal brainstem type level, and that I suppose even in humans to some extent all we're doing is modulating those reflexes and those pattern generators, but clearly in mice you can get away with that a great deal more. Okay. I've probably run out of specific questions then on the question of the motor side of things, or the specifics of the motor consequences of action. I suppose the other area that people sometimes talk about here, one more area on that I suppose that's worth briefly covering is that of sensory attenuation, and some would argue that under some predictive processing accounts, the only way to generate meaningful movement is to be able to sort of transiently suppress the predictions you've got coming in. The sensory data that's coming in that suggests I'm not currently moving, so that you can entertain a belief that I've started moving, so that you can predict that I should experience the sensory consequences of movement, so that you can then fulfill those prediction errors and start moving. And there's an argument that there are interesting arguments about how much of your sensory data needs predicting there. Is it just suppressing? Is it just attenuation of proprioceptive data, or is it much broader? is it something that also affects visual data, any consequences of action? And there's been, I think there's some data suggesting that it's much broader than that and covers other forms of sensory consequences of action as well. On that, it was quite interesting that actually you showed some slightly larger responses for, if I remember the slide correctly, in response to unexpected consequences of action versus other kinds of prediction errors. Have I remembered that correctly? I think on one of your earlier slides.
Yes, I believe that's correct. If you're referring to the fact that visual motor prediction errors tend to be much larger than other types of prediction errors, what do you mean that the consequent, if the stimulus is self-generated, it results in stronger responses than if it's passively observed?
Yeah, I thought that was interesting in that seems to go against one of those that big argument, the idea that if you're when you're generating a motor command, you're effectively suppressing the consequences of that. And people often use the example of sort of saccadic suppression and the fact that you don't see anything while your eyes are moving from one position to the next. You just sort of see the sort of broader scene rather than the motion while you're making a saccadic eye movement. And it just struck me as quite interesting that you were suggesting you actually get a larger amplitude prediction error when it's self-generated than not. I don't really have a clear explanation for that in mind, but I just thought that was notable in that sort of it's something that only comes up really in that domain of self-generated data and when you start to bring action into it, and whether that's another sort of subheading that becomes relevant when we're dealing with that sort of action. That element of predictive processing.
Yes.
Yeah, no, go ahead.
Yes, to your first point of do we need to suppress the prediction errors to induce movement? To my mind, that may not be equivalent, but at least conceptually is very similar to the idea of or to Carl's idea of precision weighting of the prediction errors. And we've actually searched endlessly for a mechanism that could implement something like precision, right? Most people speculate that it's some neuromodulatory system that can do it. And we've tested almost every one we have tools to test for in mouse visual cortex to see whether there's any gating of these prediction errors by neuromodulatory system. And the short answer is definitely not at the resolution we would need to actually do something interesting. So, I mean, we've looked at cholinergic input, we've looked at noradrenergic input, serotonergic input, and even to some extent dopaminergic, see whether any of these signals have the resolution to drive prediction errors with any degree of specificity. I mean, and what I mean here is right, the way people imagine attention working, that we can attend to this part of visual space versus that one, and then selectively gate prediction errors in one or the other. And the short answer is none of the neuromodulatory systems, they all do interesting things. I mean, locus coeruleus probably broadcasts if there's a large enough prediction error in visual cortex, you activate nonlinear the noradrenergic input that likely is relevant for plasticity. So most of these neuromodulatory systems are likely plasticity related, but don't have anything to do with precision. And I've not come across any, I mean, given what I've just said to you, I've not come across any other viable suggestion for how this could be implemented, short of having actually, I mean, an early version of, before we thought of these two layers as JEPA or a JEPA-esque implementation, the superficial and deep. Our initial idea was actually maybe the entire layer 2-3 network functions like a precision network for layer 5, such that you have a predictive processing network in layer 5, and the sole role of the layer 2-3 network is to compute the precision of all of these and dynamically readjust the prediction errors computed in layer 5. Something I haven't told you is that there's actually a beautiful symmetry of prediction errors in layer 2-3 and layer 5. So if you, sorry, maybe it's easiest if I show you the superficial. So the negative prediction errors are layer 2. they sit. Sorry, I'm just going to use this diagram here. So the negative prediction errors are in superficial layer 2-3 in the mouse. They're also in superficial layer 5. So the superficial layer 5 cells are negative prediction errors. The deep layer 2-3 cells, the layer 3 cells are positive prediction errors. The deep layer 5 cells are positive prediction errors. Actually, layer 5A, or the superficial layer 5 neurons, have even stronger negative prediction error responses than the ones in layer 2-3. So there's this weird functional symmetry that is also mirrored by connectivity. So these cells that have similar functional responses also tend to communicate more directly with each other. This is based on the Allen EM reconstruction of mouse visual cortex. And so, one idea that we had is that, I mean, this is very vague, right? But given that none of the neuromodulatory systems are anywhere close enough to have the resolution to do this, as far as we know, I mean, this might just be an experimental limitation, then maybe it's the entire layer 2-3 networks that actually does this job of modulating the layer 5 gain, if you will, or basically functions as a precision for the layer 5 predictive processing network. And this might still be true. I mean, we have no evidence against this. It's just that JETPA is an equally nice interpretation that we have a closer match for, given that layer 2-3 seems to predict layer 5, which I wouldn't necessarily have predicted based on a precision type argument. So I think, I mean, that is one of the key problems, is answering the question of how is the idea of precision, or how could this even be implemented, short of just assuming it might be some neuromodulatory signal, which it very likely is not. I mean, we haven't fully ruled it out, and actually just a few months ago I saw the first data that seemed to indicate there might be very local response types in the noradrenergic system, but the number of neurons is just not sufficient to have the resolution necessary for this. But if you really want to code at even, let's say we have 100 times less resolution than the cortical coding space, right? We can't focus on one receptive field, but 100 times less. That would still mean you need 100 times less neurons than cortex has to do this. And none of these neuromodulatory systems have that capacity.
When you're talking about resolution there and the limitations, there's essentially saying in terms of being able to focus in on really specific areas of visual space, or
well, exactly. You want to modulate the precision, or you want to activate this finger but not that one, right? And you want this one to allow it to move, or etc. Right? The resolution needs to somehow be match of the precision to the coding system that you're trying to control. And so you can allow for some mismatch and say, well, it can be a bit fuzzier, but probably not orders of magnitude less precision. What the neuromodulatory system, right? Now we're talking 5-6 orders of magnitude difference in resolution to cortex. I mean, plus most of the functional responses seem to suggest that the dimensionality of the entire cholinergic system is maybe 2 or 3, right? I mean, all the axons tend to do the same thing, and it's very, very low dimensional coding. The same is true for the serotonergic system, and presumably the dopaminergic, where I don't mean to imply it's just one dimensional, right? And people actually, the debate in the field often is it completely homogeneous, or are there domains? Which means, is it one dimension or maybe 10? I mean, either way, it doesn't have the resolution, right? Even if that number is not 100,000 or a million in mouse cortex, then it doesn't have the resolution to do this precision weighting. So yeah, I mean, I think that is one of the essential things I think the field is struggling with, to try to make headway experimentally in addressing, how could something like a precision be implemented? How could one design an experiment that a mouseologist can now perform and see, okay, this looks like it's a precision signal. Actually, point to where it could be. So.
Well, I mean, that's a really interesting point as well, and I was tempted to make precision point 4 of this sort of 3 themes that I was mentioned at the beginning, and probably it is worth thinking about based on what you've said there, and it's almost certainly multiple mechanisms in play to get the dimensionality, as you say, to sort of really focus in on anything. There is a slight distinction one could make, though, also between sort of the balancing of precision at different hierarchical levels versus focusing in on a specific set of channels at a specific level, or sensory feature. And one could imagine cholinergic, for instance, effects. I didn't realize they were quite as low dimensionality as you were saying, but one could still imagine a sort of effect of rebalancing things a little bit more towards sensory data at the expense of priors along some hierarchical structure in visual cortex, for instance. And I gather there is some evidence, at least a sort of evoked response level of greater amplitude responses with cholinergic manipulations compared to without, that might support that. But yes, in terms of focusing in on a really specific receptive field, I think you're exactly right. That there has to be other mechanisms in play, and whether that's more to do with that modulation locally in more superficial layers of cortex. But yeah, no, absolutely a really interesting and important part of this, I think, particularly once you start thinking of precision of different things and of different types. So I guess what that sort of deals with precision of the sort of predictions one might make, and therefore the weighting of the prediction errors going in the opposite direction. You can also then start to think about precision of predictions in time and think about what's going to happen next, and how volatile the world is, and that comes back to some of these points about sort of anticipating what's going to happen next and therefore making decisions based upon which of those anticipated outcomes I like the most. And you can then also start to think about precision that affects the decisions I might make, and how certain am I that I'm pursuing the right course of action. People often think about in terms of dopaminergic type modulation, but again, that might sort of lead us more in that subcortical direction. So I think from what you say, it sounds like that becomes a really important part of that sort of discussion ongoing as to what are the open questions for the field, what are the controversies and things that need to be resolved. It's sort of the way in which different precisions might be disambiguated behaviorally, but also pinned down anatomically and neurochemically.
Thomas, can I interject here? So before I forget, because there's so many really interesting and important points, I'm not going to hold them all in mind. So I'm aware this has been recorded. So one of us is going to have to go back, and I suspect Thomas is the youngest, so it's going to have to be him, but we'll see about that. Just a number of things that came to mind. First of all, precision is not really, although it's very sweet of you to say so, it's not really my idea. It's a fundament of any data assimilation scheme or sense-making scheme that so you can trace right back to things like Kalman filtering, or it's called the Kalman gain. The key point I wanted to make of several was that it is the thing that encodes uncertainty. So when we talk, so precision is not just a little add-on. It is a fundamental aspect of sense-making, decision-making under uncertainty that acknowledges you have to encode the precision. In the particular formulation, or how it unpacks when you read predictive coding as a Bayesian filter, it manifests as effectively a gain on the encoding of prediction errors, however you think they're encoded, whether it's a sort of population activity or particular prediction error cells or dendritic components. It doesn't matter. You have to have a quantity that encodes the uncertainty or inverse uncertainty of the information, if you like, subtended by that particular prediction error, however it is expressed, in play. So when people talk about precision weighted prediction errors, they're just saying that it's not just the content of the the mismatch. It's, how much credence do you afford it? How certain or uncertain are you? And that has to be encoded. The reason I'm whittering on about that is that in my again tongue-in-cheek arguments with Jan LeCun and his JEPA thing, the reason that we never quite converge is that as an engineer, he doesn't like uncertainty. So he makes his life really easy by what he would say adopting a zero temperature free energy. Me, as a physicist and a fan of Kalman, think that's just not on. You can't do that. So I've never taken JEPA seriously just because it's the wrong kind of maths. But interestingly, if you've seen a parallel, and I certainly did, I'm just thinking of Stuart Shipp's deconstruction of this second order kind of predictive processing. And by second order, I mean the second order statistics. That just is the uncertainty, encoding the salience, the reliability, the credence, the uncertainty or the precision of various sufficient statistics. At one point, he was actually proposing two streams, and it's really interesting that you also, George, you considered that there was one a sort of first order predictive processing counter stream predictions and prediction errors, and then a second order one that was literally about the second order statistics, which would be the precision, various ways in which you could encode uncertainty by predicting the precision or predicting the predictability. I think that would be really nice access to, to expose as an outstanding question, as you know, given that neuron review and our subsequent exchange. But it just struck me that that it's interesting you've ended up with JEPA, which is exactly the scheme that doesn't have this. And I'll be quite bold and will never work. It's just because it's not encoding uncertainty. So if there's somewhere to repair that dual stream notion from JEPA and the sort of Stuart Shipp formulation of first and second order counterstream, I think that would be great. The other point I want to make is I wouldn't get hung up on neuromodulators. When people like me talk about neuromodulation, we just mean a change in the conductance as opposed to depolarization. We just mean there is a second order effect of the kind associated with voltage-dependent NMDA receptors that changes the conductance, not without changing the depolarization. So the driving effect would be manifest as a depolarization, but the neuromodulatory effect would just be a change in conductance. And there are a whole raft of different mechanisms that underwrite that kind of neuromodulation. So although neuromodulators, I think, are extremely important in terms of setting the tone, as Thomas was intimating, that's not the complete story. We're talking about sort of everything from after hyperpolarization, depending on intracellular calcium, through to synchronous gain of the kind that you would get in communication through coherence with timescales, I would say 300 to 400 milliseconds. Anything that affects the postsynaptic gain or the slope of the voltage current activation function, anything that affects the conductance of a postsynaptic response will be a plausible mechanism for this kind of gain control. And it just strike me that and it comes at many different scales and many different flavors. And the reason I'm sort of pursuing this now, before I forget, is that the way that you were describing in one of the early models, where there is stuff gathering prediction errors of positive and negative side in layer 5. That is precisely the architecture that you would want to use if they were gathering the prediction error squared. Because what you can do is forget about neuromodulation from the point of view of cholinergic neurotransmissions, and just think about how I would locally estimate the precision of my prediction errors. what I need to do is just to basically evaluate the sum of squared prediction errors in the same way that when you evaluate the standard error for a t-test, you just sum locally.
Well, I think
Using
we lost
exactly the lateral columns, and you just sum them, and then you broadcast that back. So if you've got your precision estimate is locally too low, meaning that the actual data, you're thinking that the data is imprecise, and yet it is more precise, then your sum of square prediction errors is going to deviate from your prediction of 1 usually, and therefore you can then broadcast that back locally to then readjust the sensitivity so that the postsynaptic gain is locally controlled. So this is basically a local cortical gain control that just basically keeps the excitation balance in check just by summing the squared activity mathematically locally to make sure that the average sum of squared prediction error hovers around unity. Now, these mechanisms don't require anything other than some kind of conductance modulation and local lateral interactions. And this is exactly the mechanism that Raphael was working on before he got into the machine learning kind of predictive coding. So he did some very beautiful work, which he seems to have forgotten about, and I keep encouraging him to go back to, because it was a really simple and elegant way of implementing local precision control through lateral inhibition effectively, where you're just gathering the local activity of prediction error units, should they exist, and then modulating their sensitivity to afferents in order to make sure that the pooled sum of squared, the pooled overall activity, was brought back to, and mathematically that's identical to the Kalman gain. So that would be a very local one, and then you sort of get a bit more move to a larger scale, and then there are arguments that the similar kinds of mechanisms could involve interactions with the pulvina. So if you look at the sort of the cellular anatomy and the connectivity of reciprocal connections between visual cortex and the pulvina, for example, you've got all the machinery that you would need to do the same kind of thing. Nothing to do with classical neuromodulators. It's just increasingly large-scale mechanisms to keep your precision estimates, or to ensure that you have got the right kind of predictions of uncertainty or prediction. And on that view, you can start to talk about sort of representational sharpening. You can understand now divisive normalization as a local operator. So I think there's if this is framed as an important issue, I think it would be useful just to list all the possible mechanisms that come under the rubric of precision weighting as used by people like Jacob Howie and Andy Clark, or Kalman gain as would be used by control theoreticians, or attention, or all of that good stuff. I think it'd be useful just to point out you're just looking at, in the first instance, just looking at local lateral, basically inhibition. And there's quite a lot of empirical work showing that this can certainly provide a plausible explanation for what you measure non-invasively. The other thing that comes to mind is the reason I got convinced that neuromodulators were well, not me, but the field were important, was some very early work by Weinberger, Irvin, in the 1980s and 90s, showing that in fact you could get cholinergic modulation of very, very precise receptive fields in the auditory cortex. So there is evidence out there that in fact cholinergic neurotransmission actually has an extremely high dimensionality. However, notice that if you're modulating something with a very low degrees of freedom and dimensionality, if you're modulating something with a modulation mechanism that has low degrees of freedom, but the thing you're modulating has high degrees of freedom, it looks as if the modulation is very high dimensional. So that came to mind with your discussion, that because it's a modulatory effect, it's a nonlinear effect, so it's an interaction. So you can get away with very coarse-grained classical neuromodulatory cholinergic of the kind that people like Sabine Kastner has indeed spent much of her career studying. You can get away with a very low dimensional, coarse grain projections, cholinergic projections from the nucleus basalis, but their effect, the way that they are manifest, is an interaction with the stimulus bound processing that can have extremely high precision. So I just wanted to get those out of my system before I forgot it. Back to you, Thomas.
I think I'm nearly done as well. I think that the sort of final question I had was sort of coming back to that much more sort of anatomical circuit level description, and it was just to ask you, in your graphics and in the circuit you depicted, I was interested to see there was an internal representation that was connecting to the prediction area units and being updated by them, and then there were separate arrows labeled prediction. And I was trying to understand, what is the internal representation if not a prediction?
I see. Yeah, no, this is it might be our poor attempt at explaining this, but the idea is that this circuit is repeated across cortical areas. So the source of prediction if you're in V1, but you have local internal representation neurons, and those are the source of predictions to V2 and A1 and M1 and whatnot. But in V1, the source of prediction is other layer 5, other brain areas, cortical areas, layer 5 neurons, or internal representation neurons. I mean, that was the model at the time.
I see. So your internal representation neurons are also the same neurons that are then doing their predictions for the lower layers.
Yes, exactly. And then and part of the reason we moved away from or restructured the round Ballard suggestion in this way is precisely that it should work without reference to lower or higher. Whereas the round Ballard only works if you keep it hierarchical, because they moved to the internal, basically it's their internal representation neurons that we moved here are in the next layer in their hierarchy. And this separates this a bit such that it no longer works non-hierarchical.
Yes, which is another interesting issue, I suppose, in itself, in that. So you're suggesting that the errors and predictions are all bilateral.
Yes, yeah. I mean, based on the assumption that the cortex as a network is not hierarchical in any meaningful way. I mean, obviously one can define a hierarchy, but that's true for any network, like Erdős has proved. But the cortical network isn't hierarchical. It's more of an arbitrary graph.
Okay. I'll just so just in terms of the sort of, I guess there's quite a lot of work in the kind of late 20th century focused on asymmetric patterns of laminar connectivity and sort of directionality of those connections. I appreciate it might be difficult to sort of construct a strict hierarchy, but how is that sort of out of date at this point? Is there
to a large, there's multiple levels to your and multiple levels of an answer. The short one is yes, it's out of date and incorrect. The slightly longer one is there are definitely asymmetries in the visual pathway, but sort of the analogy, I think that it might be slightly obnoxious, but explains it best, is it's a bit like assuming the Earth is flat, which is an awesome thing if you want to get to the train station. If you're trying to circumnavigate it, it's a bad idea. Right? So it's not that that it locally does not look like a hierarchy. It's just that the thing globally can't possibly be.
In what way? What goes against
So
that?
if you look, what that it can't possibly be.
Yeah.
How would you? I mean, where's motor cortex?
Yeah, go on.
You cannot define a hierarchy that fits to all brain areas, right? I mean, you can, but the image is more like how do you? Oh, sorry. Let me share again. How do you take a network that looks more like this, right? Right, more like the disordered set, this, right, where there's areas connected to the retina, to the cochlea, to motor effectors, and then squish all of this into one hierarchy. I mean, that obviously, right, you can define a hierarchy, like how far away in publication space you are from Erdős, and say that's my Erdős number. which works, right? You can say how many synapses am I away from the retina, but that's as arbitrary as the Erdos number for publications. The or the Erdos index, whatever you called it, I forget. But the anatomically this isn't a hierarchy. And it doesn't work, right? So if you do use the Henry Kennedy approach to structuring the hierarchy, we can actually pull. I don't know if I can do this. I'll send you the link later because it might take me a second to. It's
Well,
actually
I suppose the question I had is whether if the asymmetric connection patterns are accurate, is that a
yes,
way of sort of, or
right,
do
right. So
you find that the same connections are actually relatively symmetrical between pairs of areas?
um, so they are locally there can be asymmetries. I mean, to quote one of the people that was instrumental in this field who's still active is Henry Kennedy. A lot of the early work is from his lab, and I recently spoke with him, and he said in private communication, the only truly asymmetric connection that they still find is between in primate the visual system is the one between V1 and M5. MT, V5 in humans, MT. And that seems to be truly asymmetric. Now, he actually published a paper in Science in 2013 called Dual Counterstream Architecture, where they've expanded this approach. So the original, Velleman-Van Esten hierarchy is all data in the visual stream of the primate. They've taken that approach and now done it cortex-wide. And they use those principles to construct the hierarchy. Now, I usually this is a figure from their own paper, from Henry's paper, that I like to show people that I was looking for, where I ask them what's at the bottom of the hierarchy, and they all say, well, V1. It's like, excellent. So where are all the prefrontal interesting areas that people tend to consider higher order areas in this hierarchy? It turns out they're all smack in the middle of their hierarchy. And then the question is, what's at the top of their hierarchy? And that's S1. Primary somatosensory cortex. So, I mean, and you just look at this, they have a much more interesting idea in the paper. I think their own data just shows this whole hierarchy idea is gibberish. But the more interesting point they make in the paper is that you have a dual counterstream architecture, that superficial layers have a bidirectional communication and deep layers have bidirectional communication. And there are biases as you get to the periphery of the network, closer to V1. There are clearly biases and asymmetries in how they connect. None of this holds as soon as you move away from the very peripheral areas. Right? If you don't look at V1 but take a random brain cortical area, none of this holds. So you cannot define a clear hierarchical relationship, neither functionally nor anatomically nor any meaningful way that will. Sorry, this is now me ranting, but I mean, there's good data to indicate that none of this is really quite as strict as we thought about. And we really, as a field, need to get rid of this Velleman and Van Esten idea of the hierarchy. I'm sure they had many good other ideas, but the hierarchy idea it belongs abolished from all of our thinking.
Well, that perhaps then needs to be almost the central point of any paper on predictive coding and its physiological underpinnings.
Can I interject again before I forget?
You can take over from here, Carl. I think
Oh,
I've
no. You need to close out. I mean, we must all be getting tired now. Poor Peter's sort of being patient and sit.
I'm enjoying myself.
Yeah, that was a really interesting exchange. I know Henry and his work. I used to be on their SAB when he was publishing that and his bow tie architecture and the like. I think this is just to reiterate what Thomas says. I think this is fundamental because George, you were saying I finished my rant on this, but it seems to be a really important rant. And certainly, I think again needs to be exposed because most people in my field. When they talk about predictive coding, they immediately in their head hear hierarchical predictive coding with multiple levels of abstraction and all that entails. Having said that, the way that you, the thing that you clearly want to rant against is a sort of Velleman and Van Essen strict subsumption hierarchy. And so what that suggests to me is I think it'd be nice to have an ontology of what people mean by hierarchy. When I think about hierarchies, I'm with Marcel Mesulam. I'm with a centrifugal hierarchy, and I'd actually call it a heterarchy now, where it's perfectly possible to have lateral interactions and have no strict ordering as in a subsumption hierarchy, and indeed not even have one center. You can have lots of epicenters in it, and that's very consistent with your diagram. But there's still in that conception that these centrifugal hierarchies that are based upon human neuroanatomy, where there's deeper stuff and more superficial stuff. So there is more sort of central stuff and more peripheral stuff, indeed, as you articulated. That's what I mean by hierarchy. I don't mean a sort of ladder, in the sense of and I think that sort of centrifugal hierarchy also came through with Henry's work in terms of his bow tie architecture. I never quite understood the focus on the bow tie architecture, but I got a sense that there was a kind of centrifugal aspect to it, which I think is a much more plausible way. So I think that it may be good just to have a taxonomy: hierarchies, heterarchies, linear, centrifugal, non. And it strikes me that what you mean is basically a centrifugal heterarchy. But then the deep question does arise, because the early work that was used, the forerunner to Henry Kennedy's work, which was done by people like Scannell and Young, and even based upon the work of people like Mounsell and the others, was all on what Thomas was asking about, which is asymmetries in the forwards and backwards connections. So you can't define any kind of hierarchy, even a heterarchy with lateral interactions and bidirectional interactions, unless, well, you can. That's if there's any heterarchical aspect to it, such that one level can be regarded as higher than in relation to another level, but not in any ordered way. But just if I take two areas, I can say this is higher than that, or that is higher than this. Then there has to be an asymmetry in the extrinsic connectivity that is usually read either in terms of lambda specificity or in terms of the preponderance of modulatory connections as opposed to driving connections in the Sherman Gullery kind of sense. So I'm tempted to ask you, but I don't think we got time to I might engage another rant if I did. I'm tempted to ask you, is that common thinking now? Forget about hierarchies and heterarchies. Is there a notion that there are different classes of interareal connectivity profiles, either in terms of the laminar specificity or in terms of the neurophysiology, of the kind you'd access with, say, Cooley experiments or optogenetics?
It's a good question. I mean, there's definitely emphasis on differences, but in a more broader context of large similarities. I mean, people do study differences, but I think it's really just on the backdrop of assuming that most are somewhat not identical, but yeah, very, very similar.
Yeah. Sorry, I was brought up in the 90s, the lateral connections, not horizontal connections within an area, but lateral connections within a heterarchy were sort of bilaminar to bilaminar, as opposed to the sort of forward connections targeting granular layers and backward connections targeting superficial layers.
Yeah.
You allow for those lateral connections, then that seems very consistent with what you're saying, that people assume a background, it's going to be a lateral connection in the first instance. But if there is any hierarchical ordering, it could be very context sensitive. I think frontal eye fields from Henry's work was something that sometimes is right low down, and frontal eye fields can be right high up, depending upon what other areas you're looking at, which of course is the hallmark of a heterarchy. I'll stop talking now, because I just think that's going to be very important, because it did worry me that you wanted to dispense with hierarchies, because that takes a lot of structure away from the putative generative models that underwrite these predictive coding schemes that do speak to different levels of abstraction. I did notice in your graphic you had association areas and then unimodal areas. So the way that that would be read by a theoretician is that the association area is in its name, or by virtue of its name, has the ability to associate more specialized peripheral areas. So that would, for me, be a centrifugal heterarchy just by name. But what's the evidence for that? And then you could say to me, "Oh, well, it's I've got connections from 3 modality specific or primary sensory areas here." So that would be one kind of
No, no,
functional asymmetry.
I think I'm on board with this. I mean, I think this might be a semantic difference or a semantic mistake on my part. I think that
Oh well,
there.
no, I don't think it's a mistake. I think it's something that we should expose because if you are reading hierarchy as Fellman and Van Nessen, that means a lot of people will, and yet a lot of
Oh,
people
yeah.
don't. So I think it's beholden to us to actually just write out the different ways in which you can read hierarchy and what commitments there are, because just pursuing that notion that if I spoke to people like Semizeki or Mansell or Pandya, Rockland, and indeed some of them I actually did speak to, they wouldn't worry about lambda specificity at all. They just worry about convergence and divergence of projections. So if you're higher up, you've got more divergent projections than if you're lower down, and your projections are narrower and more convergent, so that you get an elaboration of receptive fields as you get further and further away from primary sensory cortex. So that would be another metric, which is purely not on the lambda specificity or the physiology, but just upon the spatial divergence and convergence of extrinsic connections. I just put that there so I don't forget it, or someone doesn't forget it, as another, how do you define a hierarchy? What does a hierarchy mean? And is there a hierarchy? Should we talk about heterarchies? If there's a hierarchy, what does it mean to be higher or lower than something else? Well, there are these ways of and what's the evidence for this? I think all of these are really important, certainly from my perspective in terms of reading the architectures of the cortex and subcortex as scaffolding a generative model that actually installs the cause effect structure of the sensory motor sort of contingencies that we're studying. To my mind, that's a really important point.
Yeah, absolutely. Yeah, very good. Very unfortunately, I short-sightedly only reserved the 2 hours, and will need to leave in about 10 minutes.
But I also gathered from Carl that I think it's a good
Very good.
time probably for most of us to
Yes,
end the meeting
maybe.
here.
Oh, I'm sorry.
Soon.
Maybe in summary, I did speak to Mariella Zierlinger last week. She's the editor-in-chief in Neuron, about this, and in principle, I mean, she would be open for publishing something. What she was clear on, she doesn't want an adversarial view on this, but if there's some consensus, and I mean, in my view, I think the value, as I already said in the email, the value of doing this would be to find a common language between the fields, exactly of the nature that we now discussed, right? When I say hierarchy, I mean this, and when you say it, you mean something slightly different. In particular, with also, I mean, I truly did not realize when you explained what you meant by neuromodulation. That was quite helpful, because experimentally that is just a difficult definition, because you can't experimentally distinguish between things that change the potential and things that only change the conductance, because that just never happens. If you change the conductance, you always change the membrane potential. So conceptually, that makes a lot of sense, but experimentally, it's almost impossible to distinguish. And so neuromodulation just means something very different to an experimentalist or a circuit neuroscientist in the way you might use it. Which I think, I mean, that will probably be one of the key valuable things. The other would be to define these questions, right? How can we, as you already alluded to, right? How is this implemented? The question is how is predictive coding or processing or Bayesian inference or free energy, how are these ideas implemented or active inference in neural circuits, and what can the neuron readers, right, the mousologists of the world, what can they actually take from this and go and do in their lab, right? What experiments? And I think there are a few that one we could probably propose, or that you could propose, that would be very valuable.
Brilliant.
Right.
So who's actually going to do this then? So,
yeah, somebody I guess needs to take charge. Also, I just to point this out very strongly, I'm very happy to be involved in actually doing the work. I absolutely do not need to be listed as an author on anything we end up doing. So just to be,
I think you absolutely do.
what?
that is very
From my
very
perspective.
kind of me. I see.
Yeah, I think the whole point is that if we could co-author something from theoretical versus you know the empirical or experimentalist perspective. Not demeaning your theoretical contributions, nor impugning my own empirical brain imaging work, but we do come at this from very different angles, and I think having all three of us as co-authors is a much stronger statement than and certainly it's not going to be adversarial. We just need to expose the issues for conversation and for on both sides of the theoretical experimentalist fence. I've learned a lot about we haven't had time to talk about it. I was particularly fascinated by the sort of the typing of the different inhibitor interneurons, and I mean that's a level of detail I just didn't know existed. It's fascinating. So I was thinking more practically. I'm mindful that both of you, well, I guess we're all very busy, but practically, how would we put together something? I mean, we've got already that original neural, which was purely theoretical, but it really did not address the issues which have come up during this conversation. I'm hoping that Thomas was going to volunteer to put it all together, or that George would say, "Well, actually, I've written something. I just need to endorse my theoretician." But I'm not going to say anything else.
I mean, I'm sure we can come up with a plan. I suppose the first step is really just having a sort of good selection of headings that we can sort of
Yeah.
work out what would come under it from each perspective, and then see if we can synthesize that.
Perfect. Like that. Yeah.
But why don't George? Why don't you and I sort of email offline about that, and we can sort of work out what the right way forward is.
Very good. Yeah, let's write the skeleton for what one could discuss, and then sort of fill in the detail. Once we agree on the skeleton, I think from there it's relatively straightforward.
Yeah, and I think we've probably got most of that from this discussion, but it may require watching back just to sort of pin down all those points.
Excellent. Sounds wonderful. Thank you so much for your time, and once again, I apologize for being an irritating reviewer.
No, don't be silly. And thank you. You've put in the vast majority of the work for today, so really appreciate that.
It was my pleasure.
Should we give you a last round of applause, Georg? Thank you.
Thanks a lot.
I'll send around the recording once it's ready, and it was a pleasure to be around. Even
No problem.
okay.
Bye bye.
Bye-bye.